When there's a threat to human welfare, it's met with a strong response. In this case, scientists at the Danforth Plant Science Center in St. Louis are on the front lines fighting for the future of the global food system. They are combating a disease that threatens one of the world's key crops, cassava.
So everybody knows about corn and rice. Those are the crops that we're really familiar with here in the U.S. Cassava is just as important as those crops. It's a source of starch for millions of people, but it's primarily grown and eaten by people who live in the tropics, so Central and South America, Sub-Saharan Africa, and Asia.
Cassava research in the Becky Bart Lab at Danforth is uniting scientists globally as farmers may suffer huge yield losses.
It is susceptible to a couple of diseases, one of which is cassava mosaic disease. This is a DNA geminivirus pathogen of cassava that infects the cassava plant. It's transmitted by little white fly insects.
It's a DNA virus which will get into the plant cell. It hijacks the plant's DNA replication system in the plant cells and uses that for itself, and then it will replicate very, very quickly. It can be really devastating. You could lose 50 or more % of the yield.
Danforth associate member and principal investigator Dr. Nigel Taylor has been studying cassava for 30 years. Crops infected with cassava mosaic disease don't fully develop the storage roots that are used for food. The starchy root crop is rich in carbohydrates. It's one of the most important staple crops in the world, feeding nearly 1 billion people.
Cassava mosaic disease does not yet exist in Latin America. If it gets to Latin America, it will be a huge problem because none of the varieties grown in South America and the Caribbean, none of them have resistance to cassava mosaic disease.
But research in the Becky Bart Lab at Danforth is part of a collaboration that now has the world a step closer to the possibility of planting disease-resistant crops.
So our discovery is the gene in cassava that protects it from this virus. So there's a lot of different varieties of cassava out there. Some don't have this specific gene and are therefore susceptible to the pathogen. If they have this certain variation, this mutation in this gene, then they are resistant to this disease, just genetic resistance.
The gene that we found the mutation in, it's called DNA polymerase. And it's a really interesting protein because it's part of a complex that replicates all the DNA inside of our cells.
For the discovery, research associate Carrie Gilbert used big data analytics.
Folks around the world have been working on the problem of cassava plants and this virus for a long time, and they've generated a lot of data. And so I was able to access this publicly available data, sequencing data, disease data, and look to see which plants are resistant and maybe do they have the same mutation. And we did.
The work sheds light on how to maintain resistance.
You might have one cassava plant that's really, really productive, produces huge roots underground. That's what people eat, is these storage roots, but happens to be susceptible to this disease. So you can cross that variety of cassava with a variety of cassava that has the resistance and get new varieties of cassava that combine those two traits.
The discovery offers hope for other major crops experiencing attacks by this family of closely related viruses.
Geminiviruses, like the one that causes cassava mosaic disease, are very common. They infect lots of different crops, cotton, tomato, grape, a lot of other ones as well. And in many of those cases, there are no known disease resistance control strategies. So what we're interested in doing now is figuring out whether or not we can take our discovery from cassava and recreate it in some of these other plants to generate a novel source of resistance.
This discovery could be made to operate in some of the most important crops in the world, such as cotton, maize, beans, tomato, to improve the livelihoods of hundreds of millions of people